Hemoglobin is our respiratory pigment. It's kind of a cool thing. Let me use a different color here. So let's use green. This is my respiratory pigment. So its major function is that it must bind reversibly. So that means it picks it up and lets it go to oxygen. So it picks it up in the lungs, for example, and then it lets it go in the body tissues where it is needed. This letting it go has a name, which is called dissociation of oxygen from hemoglobin. So when we look at hemoglobin, we could say that this is hemoglobin, this is oxygen. It goes in two directions. So we have oxyhemoglobin, right? And then it can release it. And so this process of releasing and going in this direction and back towards HB and O2, let's use a different color, this would be the dissociation. Okay, so it binds it and it picks it up. When we look at the structure of the hemoglobin molecule, it has a heme group and then it also has globins. So when we look at the heme group, it has nitrogen, which is kind of important. So here we see this is the actual heme group right here. It has nitrogen atoms, and then it also has iron. And the iron is elemental iron is Fe. So that's my iron. So the heme group is what binds them. So there's 4 hemes, and each can bind one O2 molecule. So when we say that the hemoglobin is saturated, or we talk about hemoglobin saturation levels, and you might have heard about this big time because of COVID, because one of the characteristics is that the hemoglobin saturation levels can fall really low, like 70% maybe, and people don't even realize it. And then they're in respiratory distress. So hemoglobin saturation has to do with the percent of hemes that are bound to oxygen. So if it's 100%, then all 4 of the hemes have oxygen. If it's like 50%, then maybe only half of them have oxygen. So that is what hemoglobin saturation levels refer to. And you don't actually want them to fall much below 90. And they do this, they measure it in the doctor's office, for example, or you can buy these pulse oximeters from Bimart or wherever you go, and you put it on your finger and they can measure your hemoglobin saturation. We also have the globin groups, and the globin groups actually differ depending upon whether or not you're an adult or a fetal. So we'll talk about the adult version first, or if you're a fetus. The adult version has 2 alpha and 2 beta globins. And so these are Greek letters. So the globin chains are right here. They are the globular proteins. And then beta globins up here. Okay. So these do not function directly in binding of oxygen, but in like sickle cell anemia, it's actually the beta globin molecules that are deficient, and that is actually what causes the cells to sickle in shape. When we talk about fetal hemoglobin is different. It has 2 alpha, and instead of beta, it has 2 gamma. So we'll put no beta globins. And you might ask, well, what is the significance of this? This is very interesting because this means that fetal hemoglobin has a higher affinity to oxygen than adult hemoglobin. And the significance of this means that the fetus can pick up oxygen from the placenta. So when the blood gets to the placenta, it's under low oxygen and it should release it, right? And the mother releases it. But if the fetus had the exact same type of hemoglobin as the mother, then it would also not pick up the hemoglobin because it would be under a condition, an environmental condition, where it needs to release it. So that's why fetal hemoglobin needs to be different so that it picks it up when the mother's hemoglobin is releasing it. And this actually is an issue because upon birth, the newborn must replace all of its fetal hemoglobin. With adult hemoglobin. And that is interesting because that means that they have to break down all of their red blood cells and replace them. And sometimes when they break down all their red blood cells, they release a chemical called bilirubin. And the bilirubin actually is a very common cause of jaundice in newborns. So as they break down this bilirubin, start a new page. As they break down the hemoglobin, bilirubin is released. This causes jaundice if the liver is not able to deal with all of that. And this is a yellow coloration of the skin. How can you treat this jaundice? You could actually put them underneath UV lights or take them out into the sun. So UV lights or sunlight, sometimes they call these UV lights Billy lights, can help to break down the bilirubin.